All right, everyone, let's get started. Today, we are diving into one of the most fundamental concepts in all of oncology, the hallmarks of cancer. Now, you know there are hundreds of different types of cancer affecting all sorts of tissues, but what's absolutely fascinating is that they all, in one way or another, follow the same playbook. They acquire a common set of skills that lets them grow, survive, and spread. If you can understand these hallmarks, you're looking at the actual blueprint for malignancy. So let's jump right in. Okay, first things first, let's get our terminology straight. The word you see here is neoplasia. It literally just means new growth, and it's the proper term for what we call cancer. At its absolute heart, cancer is a genetic disease. It all starts with damage, with mutations to a cell's DNA. And not just any mutations, but specifically mutations in the genes that control the most basic cellular jobs, how a cell grows, how it repairs its own DNA, and how it knows when to die. Everything we're going to talk about today stems from these initial genetic screw-ups. So here's our game plan for today. First, we're going to define what we mean by cancer's operating system, this whole framework of the hallmarks. Then we'll get into the real nitty-gritty, breaking the hallmarks down into three main groups: how they achieve uncontrolled growth, how they become immortal, and finally, how they learn to hide and spread throughout the body. We'll wrap things up by connecting all this biology to the real world with some key numbers you need to know.
So let's talk about this operating system for cancer. I want you to imagine it's like a piece of malicious software that gets installed into a healthy cell. This corrupt code completely rewrites the cell's basic rules. A normal cell is a team player, right? It listens to signals, it knows its place. But once this new operating system is installed, it becomes a rogue agent that ignores all the normal checks and balances. We're going to go through this reprogramming step by step.
Okay, let's dive into our first group of hallmarks, and this one is really the big one. It's what most people think of when they think of cancer, just out of control, relentless growth. Your normal cells are polite. They wait for a specific signal from the outside before they'll even think about dividing. A cancer cell? It learns to completely hijack that process, creating a situation where the growth signals are always on. Let's see how they pull that off. Okay, this is probably the most famous analogy in all of oncology, and for good reason. Think of a car. In every normal cell, you have genes called proto-oncogenes. These are the good guys. They're the accelerator pedal. They tell the cell when it's appropriate to grow and divide. But a mutation can turn that proto-oncogene into an oncogene. And that's like the accelerator getting floored and stuck to the mat. It's just constant go, go, go. On the other hand, you have tumor suppressor genes. And just like the name says, they're the brakes. Their job is to stop cell division if something's wrong. So mutations that knock out these genes, that's like cutting the brake lines. To get a really bad cancer, you almost always need both, a stuck accelerator and no brakes.
So how exactly does the cancer cell jam that accelerator pedal down? Well, it has a few tricks. First, it can create an autocrine loop. Auto means self, so the cell literally starts making its own growth factors and then responding to them. It's talking to itself in a feedback loop, telling itself to grow. Second, it can mutate the receptors on its surface. A classic example is HER2 in breast cancer. The receptor itself gets stuck in the on position, so it's screaming grow even if there's no growth factor around. And third, it can mutate the messenger molecules inside the cell. A really common one is RAS. When it's mutated, it's like a faulty switch that's permanently on, constantly passing that divide now message down the line.
So we've got the stuck accelerator. Now we need to cut the brakes. This is all about becoming insensitive to anti-growth signals. Normal cells have proteins like RB, which is a major gatekeeper that can stop the whole process of cell division. Cancer cells just mutate it and take it out of commission. They also learn to ignore signals from their neighbors. See, normal cells, when you grow them in a dish, they form a perfect single layer and then they stop. That's called contact inhibition. Cancer cells? They lose this. They just pile on top of each other, forming a chaotic mass. They do this by getting rid of the adhesion molecules like E-coherent that let them feel their neighbors. Now, all of this crazy division, it takes a ton of fuel and a ton of raw materials. And this leads to this really fascinating change in metabolism called the Warburg effect. Now, this is a key point, so listen up. Even when there's plenty of oxygen, cancer cells switch to a less efficient way of making energy called glycolysis. Now, you're probably thinking back to your biochem classes. Wait, that makes way less ATP. Why would it do that? Well, it's because the cancer cell isn't optimizing for energy. It's optimizing for building blocks. Glycolysis provides all the little carbon skeletons it needs to build new lipids, new nucleotides, new amino acids, all the parts for brand new daughter cells. It's all about manufacturing speed.
All right, so our rogue cell is dividing like crazy.
But, that's actually not enough to make a lethal tumor. Normal cells have two really important safety features built in. One, if they get too damaged, they're programmed to self-destruct. And two, they can only divide a certain number of times before they just stop. So for our next section, we're going to see how cancer cells get around both of these problems to become, for all intents and purposes, immortal.
First up, it has to figure out how to cheat death. And I mean that literally. We're talking about apoptosis, or programmed cell death. This is an absolutely essential self-destruct sequence for any cell that has too much DNA damage, and the master controller of this whole process is a gene called TP53. It's known as the guardian of the genome. When things go wrong, TP53 hits the self-destruct button. So what do cancer cells do? They mutate TP53. They take the guardian offline. By doing this, they can survive and keep dividing, no matter how messed up and unstable their DNA becomes. Okay, so the cell has disabled its self-destruct button. But there's another problem, a natural lifespan. Most of our cells can only divide about 50 or 60 times, and that's it. It's called the Hayflick limit. And the reason is, every time a cell divides, the little protective caps on the ends of our chromosomes, called telomeres, get a tiny bit shorter. Think of them like the plastic tips on a shoelace. Eventually, they get so short the cell just stops dividing. Cancer's solution to this is just brilliant. In about 90% of cancers, they turn back on an enzyme called telomerase. This enzyme's only job is to rebuild those telomeres, making the chromosomes forever young and allowing the cell to divide an infinite number of times.
Okay, let's recap. We've got a cell that's growing endlessly, and it's immortal. It's a pretty scary thought. But to become a real clinical threat, a tumor has to solve a few more logistical problems. It needs to get a blood supply, it has to learn how to hide from our incredibly powerful immune system, and ultimately, it needs the ability to pack up and move. This brings us to our final set of hallmarks, hiding and spreading. A tumor can't just grow indefinitely on its own. It's like a tiny city that runs out of supplies. Once a tumor gets bigger than about 1 or 2 millimeters, the cells in the middle start to starve for oxygen and nutrients. To get any bigger, it has to trick the body into building in its own personal blood supply. This process is called angiogenesis. And tumors do this by pumping out signaling molecules, the most important one being VEGF, vascular endothelial growth factor. VEGF is like a magnet for blood vessels, telling them to grow new branches right into the heart of the tumor, feeding it everything it needs to expand. So this raises a really important question, one that your future patients will benefit from us understanding. We have an entire immune system, an army of cells whose job is to find and destroy abnormal cells like this. So how on earth does a tumor survive? How does it fly under the radar or even fight back against our own defenses? Well, they have some incredibly sneaky tactics. First, they can put on an invisibility cloak. All your normal cells constantly show little pieces of what's inside them on their surface using a molecule called MHC class 1. It's like showing their ID card to patrolling T cells. Some cancers just stop making MHC class 1, so they have no ID to show. They become invisible. Second, they can release chemicals that suppress the immune system, like TGF-beta. But the third trick, and the one that has changed modern medicine, is hijacking what we call immune checkpoints. Now let's zoom in on that last point because it's a huge deal. Think of this interaction here, it's like a secret handshake between cells. Your T cells have a receptor on them called PD-1. When that PD-1 receptor shakes hands with another molecule called PD-L1 on a healthy cell, it's an off signal. It tells the T cell, hey, this guy's friendly, stand down. So what do crafty cancer cells do? They cover their own surface with PD-L1. So when the T cell arrives ready to kill, the cancer cell gives it that secret handshake and basically tells it to turn itself off and go away. And here's the amazing part. Once scientists figured out the secret handshake, it led to an absolute revolution in cancer treatment. The drugs you see on this table are called immune checkpoint inhibitors. They're basically antibodies that physically get in the way of that handshake. So a drug like pembrolizumab will stick to the PD-1 on the T cell, so the tumor's PD-L1 can't shake its hand. The off signal never gets sent. We call it releasing the brakes on the immune system, letting our own T cells wake up and do the job they were trained to do. It was a Nobel Prize-winning discovery for a reason. All right, we've come to the final and frankly the deadliest hallmark of them all, the ability to invade and metastasize. This is what turns a local problem into a body-wide disease. It's a multi-step process, like a prison break. First, the cells have to loosen up. They get rid of that E-coherent we talked about so they can break free from the main tumor. Step 2 is invasion. They release enzymes that chew through the surrounding tissue and basement membrane. Then they have to actually move, to crawl through that matrix. And finally, dissemination. They find their way into a blood vessel or a lymphatic vessel and ride it like a highway to a distant part of the body.
Okay, we've been deep in the cellular trenches, looking at genes and proteins. Now let's pull back. Let's zoom all the way out and look at the big picture for the human population. We're going to look at the epidemiology. These numbers are crucial because they tell us which cancers are most common, that's incidence, and which ones are the most deadly, that's mortality.
So if we look at the numbers for men, you'll notice something really important. The most common cancer, the one diagnosed most often, is prostate cancer. But look at the mortality column. What's the #1 killer? It's lung cancer. This is a critical take-home point. The most common cancer is not always the deadliest. Incidence does not equal mortality.
And you know what? We see the exact same story when we look at the data for women. The highest incidence by far is breast cancer. But again, look at that mortality column. The #1 cause of cancer death in women is lung cancer. This is a fact that still surprises a lot of people, and it's a super important public health message about just how lethal lung cancer is for everybody.
But now, look at this. When we talk about kids, the whole picture changes. It's a completely different landscape. You don't see the big cancers of adults like lung, breast, or colon cancer. Instead, the things that top the list are leukemias, cancers of the blood-forming cells, and tumors of the central nervous system. This just reflects the fact that different cells are growing and dividing during development, and the genetic mistakes that happen are fundamentally different.
All right, let's bring it all home. This table is your high-yield summary of everything we've talked about. We started with getting their own growth signals by turning on oncogenes, then ignoring stop signals by knocking out tumor suppressors, cheating death by getting rid of TP53, becoming immortal by turning on telomerase, then getting a blood supply with angiogenesis, hiding from the immune system with checkpoints, and finally, learning to invade and metastasize. That is the complete playbook. And the key thing to remember is that every single one of these is a potential target for therapy.
So there you have it, the blueprint, the core set of principles that turn a normal cell into a malignant one. And as we've seen, mapping out this blueprint is exactly what gave us incredible therapies like immune checkpoint inhibitors. Which leaves us with the final, crucial question that's going to drive cancer research for the next decade. Now that we have this map, what's the next hallmark we're going to learn how to shut down? That's all for today.